like to get this webinar started at this time. So I want to welcome all of our attendees to the Windshire Group's Ask the Expert series, Pichia for Biopharmaceutical Manufacturing with our special guest, Dr. David Blum. My name is Jen Bardsley, and I am serving as the moderator for today's webinar. I currently serve as the account manager for the Windshire Group. During our time together, our panelists will provide their experience and insights on the background of the use of Pichia in its application in biopharma. Please feel free to submit questions throughout the presentation using the Q&A button in your control panel. If we run out of time to answer your questions at the end, we will respond to you directly. And if Dr. Blum, if you want to pull up the presentation. Excellent. And you can go to the next slide. And here, this is the Windshire Group. I'm going to introduce you to one of our panelists, Dr. James Blackwell. Dr. Blackwell is a president and principal consultant for the Windshire Group. Founded in 2011, the Windshire Group accelerates development and manufacturing for commercializing therapies and provides quality expertise. Dr. Blackwell has been a leading industry consultant for more than 15 years. Now it is my pleasure to turn the presentation over to Dr. Blackwell.
Thanks, Jen. And as you can see, we provide consulting services and laboratory support and consulting services to the industry, and we've been around for over a decade. So now I'd like to introduce, next slide, Dr. David Blum. He's someone who's been a industry colleague and friend of ours for a number of years now. He is director of the Bioexpression and Fermentation facility at the University of Georgia, where he obtained his PhD in biochemistry and molecular biology. So he's come full circle, where he did research on the biochemical and genetic characterization of enzymes in plant cell degradation. Then he joined Diversa, where he's a contemporary of a couple of dear friends of mine I knew from graduate school, again, where he continued research in that related area. After that, he joined the Vanderbilt University Medical Center and focused on mammalian protein expression and antibody discovery. And now he's back at the University of Georgia. Next slide, please. So I'd like to get an overview of host systems and just share a few thoughts on this. So if we look at these, historically, I think within the biotech industry, mammalian cultures have been the dominant one, expression, the host system, because of their ability for post-translational modifications that are human-like or fully functional from a human standpoint. And the productivity of these systems has increased tremendously during my careers, to the point where, from a COGS perspective, it's not a major consideration at this point, but I still think that COGS is going to be a continued driver in importance to drive those costs down in our industry. Bacterial and yeast cells maintain a lot of similarities, I think, from a production standpoint, although yeast, I think, benefits from producing less more innocuous types of products in bacterial systems. Insect cells are very common now as transient expression type of system, but they, like mammalian cells, are subjected to mammalian type of viral infections, and they have less of a regulatory history. So if I had to wave a magic wand and pick one of these that could do everything, it would definitely be yeast. And within that, it would probably be Pichia because of their more extensive history and application within the biotech industry. So with that, I look forward to learning more and like to turn the presentation over to Dr. Blum.
Thank you, James. So as was mentioned, the talk is going to focus on Pichia for biopharma manufacturing. I'm at the University of Georgia, which is in Athens, Georgia. We're about 70 miles east of Atlanta, and our university is mostly known for football, but hopefully I'll convince you that we can do a little bit more than that. So my group is the Bioexpression and Fermentation Facility, or the BFF, as we're known. And we focus in several areas where you could consider us a contract manufacturing organization that's within the university. So we're offering fermentation research. So our capability goes up to about 900 liter scale. We have a range of other equipment, including protein purification equipment. A lot of our projects focus on production of proteins, as well as creating constructs. We also have groups focused in cell culture, as well as high containment culture. Here at the bottom left is our Animal Health Research Center, which has a number of biosafety level 3 labs. We also have interest in monoclonal antibodies. My background at Vanderbilt was focused on discovery of novel antibodies, and my lab, which was led by Dr. James Crow, developed the EvuShield product, which is offered through AstraZeneca for COVID treatment. So wide range of areas, but today want to talk about Pichia. So Pichia, or Pichia pastoris, it's a methyllotrophic yeast. It's widely used in industry, as Jane mentioned, for production of recombinant proteins. So why are people interested in Pichia? A lot of it has to do with the ability of Pichia to grow to high cell density. We have observed upwards of 300 to 600 ODs, which is quite high for fermentation. I mean, the culture gets really thick. You can make upwards of 20 grams per liter of protein, which is very impressive for a recombinant organism. And it can be used to make a wide range of pharmaceuticals, RBD, antibodies, and other proteins, as I'll mention in a minute. The advantage is the, COGS are much lower compared to HEK cells or CHO cells, but it does have the potential to have even higher yields than the traditional mammalian CHO cells. So I think a lot of people haven't really paid attention to Pichia, and I think that they should. And as I'll mention in a minute, there are new strains now that can offer alternative glycosylation patterns. And these strains and technology was originally developed by the RCT Corporation. And we work with another company called Biogrammatics, who are involved with creation of strains. They specialize in strain generation. They're based out of San Diego. I can connect you with them if there's interest. And our role is basically to take those strains and then scale them up. And I'll explain how we do that in the coming slides. So as far as the biology, so Pichia, the molecular biology is going to be similar to other organisms. So you would clone in your gene. Typically, this can be done with a secretion signal. Some proteins work better as intracellular expression, but when we recommend work, we do recommend that you try at least a secretion signal like the alpha factor. And then obviously you can add on other tags, like in this example of the PPIX-Z system from Invitrogen. And then, like I mentioned, the yields get quite high. The actual expression is driven by the alcohol oxidase promoter, and that promoter is induced by the addition of methanol. So methanol also serves as a carbon source, and you can get, like I said, a secretor intracellular expression, and the proteins express are wide-ranging enzymes, antibodies, so on and so forth, as shown in this slide. This, I don't expect everybody to read everything here. This will be recorded. You can look at this later. This is also available through the RCT's website. This just gives you kind of a feel for the proteins and vaccines that are available to be made via Pichia. So you can see both, industrial enzymes, vaccines, human serum albumin, insulin, other proteins. And then most recently, there's a group out of Baylor that have produced RBD in Pichia. That's now being produced at CMO in India as a potential vaccine candidate. So a lot of potential for Pichia. One of the big disadvantages for working with yeast is that they typically will hypermonosylate. And so this can cause a big problem as far as the functionality of your protein. We have our complex carbohydrate research center. They study glycosylation, and we know the importance of glycosylation for protein expression and protein function. And so RCT has developed these strains that they call glycoswitch. And so these strains are going to have glycosylation similar to human based protein. So they're going to have your core structure, GLICNAC, MAN5, and then you have other iterations of that, as you can see in the other 4 strains. And so these are also available via biogrammatics. The strains can be licensed directly from RCT. And having this type of glycosylation could potentially be a game changer as far as production of antibodies and other proteins that really weren't possible before in a wild type Pichia strain. So then so we're a fermentation facility. So how do we produce Pichia? So essentially there's a lot, just like with any fermentation process, there's a million different ways you can do this, but we typically use a stepwise fed batch process. So we start with a basal salts and trace metals media, and then we have 2 stages of feed. So the first would be a glycerol feed, and that's typically done for a few hours or maybe 24 hours. And what that's going to do is that's going to get up your biomass. And so at the point where you have a high biomass, then you can start your methanol feed. And so the methanol can be fed in a linear fashion. You can also use methanol sensors. So Raven Biotech sells a methanol sensor that can be installed inside of your head plate or side of the fermenter to detect the methanol. And then you can create your feedback loop so you can then maintain a constant methanol supply. Because you don't want to feed in too much methanol because that could potentially be detrimental to your cells. You want to keep an even amount of methanol. So some amount of process development or using sensor technology is really critical to have an optimal process. And in the facility, before we go to a larger system like this is a 20-liter working volume system. We would then beta test that in either our DOS Gibbs system or a number of our other benchtop systems. And so that we always recommend doing a technology transfer and a small scale test before we go up to say 800 liters. The other thing that we like about Pichia is the downstream processing. So with E. coli, for instance, we're just talking about microbial systems. If we want to produce a protein in E. coli, for the most part, the protein will be expressed inside of the cell. So we would have to harvest the E. coli, then use a homogenizer to break those cells, then get rid of all of the unbroken cells and debris. It's a very long process. It also limits our ability to scale up because of our centrifugation capacity. So we use a tangential flow filtration system. And so this is a skid. We have 5 to 10 square meters of membrane. And so as you can see in the figure on the right, this is from a video that was on our YouTube channel. So if you do a Google search for UGA Master Biomanufacturing Program, which is where we teach this, you can find all of these videos. So this is our TFF video. And so if you're not familiar with TFF, I'll just briefly go through that. So you have your filter here on the right, and you have a feed reservoir here. And so your feed is pumped into the membrane. And so that membrane can be different sizes. So when we are separating cells from our product, assuming it's secreted, we use a microfiltration process that works with our membranes are 1,000 KD membrane. So that will allow the protein to transit through the membrane. And then we're going to collect the material that is being retained. That's going to go back in the feed reservoir, and then the permeate is going to be what we collect. So that will be all the protein that's now separated from the cell material. And so the process typically works this way. And our workflow would essentially be to use that that material that then transited through the membrane, and we do a UFDF or ultrafiltration diafiltration step using, say, a 10 KD membrane or whatever size is going to retain our protein of interest. And so these are just some examples of what those TFF skids look like. This up top is a sheet style membranes, and then on the bottom is a hollow fiber system. And so these are typically at the scale we run are done on skids with larger pumps. We typically get 50 liters per meter squared per hour. And so we can run through a large amount of material fairly rapidly. So that UFDF step is where we can take the product, concentrate it, and then that can either be shipped to the client, or we can use that for other downstream activities like chromatography. And then one interesting thing about Pichia is that it can make a green pigment. So if you start growing this and your culture turns green, no, you haven't got anything contaminated. These are crystalloids of the alcohol oxidase protein that is obviously part of the expression system. So that's kind of a basic overview of Pichia. I did want to mention that if you want to dig deeper into this topic, we do offer an introduction to biomanufacturing. This is through our continuing ed group that will offer this. Anyone can take this course. It's, I believe, $150, and it's a go at your own pace course. You get CME credit. We also offer a quality basics in cell and gene therapy. And this is kind of outside of what I talked about today, but that's another course we offer. And then if you really want to dig deeper, we have our master biomanufacturing program. So you can go to this website and check that out. And then this is my contact information here. And then again, I'd like to thank you for your time. This is our address, and then how to get in touch with James and the rest of the Windshire group. So we'll stop there, and I'll answer any questions that are coming up in the chat.
Thanks, Dr. Blum. One question I have, one of the limitations that I've seen for working with Pichia is the use of methanol. And so at larger scales, you'd be required to have an explosion-proof facility. Could you speak about what your limits are in terms of handling methanol are in your facility, and what you've heard from people on this issue within industry?
Right. Yeah. So our facility is actually explosion proof. We have basically a wall that would blow out if that did happen, but we do work closely with our chemical safety group. And so we have a certain limit to methanol per square foot. And so we never go below that amount. But, you do want to keep your rooms cool. The methanol that we have is, we've never had an issue with that, but that is definitely something, if your facility has any sort of flame or anything that could potentially ignite the methanol, you have to be really careful. But we have not had any problem working with methanol in our facility.
What's the potential of using other substrates?
I mean, there are other promoters systems, and I mean, they do work to some degree, but the AOX promoter has been the most yield that people have seen. So it's typically used. You could potentially make a constitutive promoter system. That's another option, but you still would need a feed. So you could potentially develop, say, a glycerol fed system, and then have a constitutive promoter. But the yields may be lower than with the AOX promoter. So you have to balance that, any concerns for safety with the product yield. So
sure, but I think compared to CHO though, it would still be pretty competitive with an alternative expression system.
Yeah.
Okay, I see we're starting to get some questions.
Yeah, so there's one about the other commercial therapeutics. So I'll go back to that earlier slide. Whoops. So this is a list of other proteins. So we have anti-RSV, antibody fragments, anti-IL-6 receptor, interferon alpha 2B, hepatitis B vaccine, and then the RBD, COVID RBD is now being manufactured as a vaccine candidate in Pichia as well. And really it's kind of up to the group. I think in general, people have sort of stuck with CHO. A lot of people are creatures of habit. They're going to use what the other guy down the road is going to use. But I do think Pichia, may be something that people should consider in the future, especially if you're trying to make things like vaccines that have maybe a low cost of good threshold.
All right, great. We have another question. What is the growth rate of these M5 strains? Is there any difficult to grow them at high density cultures?
I've not heard of any issues with growing them at high density. They typically have just one or two knockouts or knock-ins to create the glycosylated protein. So I don't think that will affect the growth rate in a major way. We've worked with the wild type Pichia, so we don't have as much experience with the glycoswitch strains. We are going to announce a new project soon, and where we're going to be exploring these strains in more detail. So we'll have more data as we go through that project. But we, definitely RCT or biogrammatics would have other information.
And what would you expect for a growth rate or doubling time, Dr.
I mean, typically, it would, we grow these in nutrient limited, but, say an hour or more doubling time if they're growing at, say, a maximal. I mean, you're not going to get like E. coli, you can get fairly decent doubling time. So, but generally, you want to get this to a high cell density and then start feeding it. And you don't want it to grow too much when it's making protein. So.
Excellent. And what is the best concentration to perform the cellular disintegration?
The best for it, I guess it's meant what's the best? Let me go. The best disintegrate, I guess they mean homogenization or cell lysis. If that's the question, then we would lyse the cells at about 5 grams of wet weight cells, 5 mils of buffer per 1 gram of wet weight cells is typically our cell slurry if we're doing the homogenization. These Pichia are tougher to lyse than E. coli, so typically 1,000 bar is good enough for E. coli, we would need to go to almost 2,000 bar for Pichia. And then I'm seeing a question about with your downstream processing, do you observe side products that are not proteins? We do see the crystalloids, so but that, I mean, that's a protein. I mean, typically there would just be whatever was in the media, and these are the media we use is a very minimal formulation, so we're not going to see a lot of side products. A lot of the biology is shunted to producing protein and not other fluxing of, you know, organic acids and things like that. And then I see another question for protein production. How much improvement can be expected by changing transcription versus secretion? I think in general, it's going to be very protein dependent. So if you, we typically like to secrete just because it's so hard to lyse the cell, especially a large volume. If you're, you know, making a lot of protein, then you really want to have it secreted. And so that's where you're going to see, you know, upwards of 20 grams per liter. And then I see, what are the major drawback compared to E. coli and CHO other than using methanol? I mean, really, with E. coli, it's easier to create the strain because you can just do a transformation and you got the strain the next day. With Pichia, you have to insert the gene into the chromosome, so there's some selection and screening, but that's also where you can find better producers. With CHO, I don't, you know, if you're making a traditional stable CHO, you know, I would anticipate CHO might be better in general at producing antibodies just because there's so much more work that's been done. But I really think that, you know, Pichia could be a system where we could explore antibody production. But I do think CHO has a bigger history, obviously, with biopharma products. Pichia traditionally have been used to make industrial enzymes and things of that nature, which it does quite well. And so not as much work has been done looking at making things like antibodies and other biopharma products.
Dr. Blum, you may recall that Glycophi was technology came out of Dartmouth, you know, created yeast that produced fully human glycosylated proteins. And Merck did a very large deal on that. I couldn't believe what they paid for that technology, but nothing ever came of it. Could you compare and contrast that experience with the current state of Pichia?
I think, I mean, with the, I'm not as familiar with Glycophi, but I would say that with Pichia, there's a lot more potential because of the ability to get to high cell density and the very prolific promoter and the AOX promoter. You know, perhaps with the Glycophi, they just didn't get the product yields they were hoping that they would get. Maybe, you know, whenever you look at companies when they're advertising, they always pick the best protein. And so if the system can't get you to the really high, you know, numbers you want, then it's not really viable. And I would expect that that's what happened. In general, we get really good yields for a lot of proteins and, you know, in our Pichia system.
I'll take the first stab at the next question, Dr. Blum, but feel free to weigh in. Is microfiltration preferred over centrifugation for culture clarification? I think it really depends on scale and economics, because in terms of economics, it is a single-use system, right? So you have, you know, those costs associated with that. But centrifugation is fixed costs. So as you scale up and get to larger scales, the economics can improve for centrifugation. And I think that's probably the main driver. And if you get at really, really large scales, it's probably easier to deal with a centrifugation type of system. But I'll turn it over to you, Dr. Blum,
Yeah.
for additional comment on that.
So for us, we use reusable membranes, and they are quite expensive. So for that 5 square meter membrane, you're going to pay about $20,000. And so we do try to reuse those. There are single-use TFF systems. So if you are running a GMP process, you would want to go with that. The centrifugation, if you know, I would definitely want to use a distack system instead of, we use a tubular bowl system, which gets a much, you know, less water content in our cells, but has limitations in the amount of cells we can collect. But using a distack system, coupled with perhaps some sort of a depth filter, and then going to your UFDF may be another option if you don't have a big TFF skid. So it would really depend on kind of what you have already, but you know, we feel like the TFF skid has a very good throughput. It's also easier to expand. You just add more membranes, you can increase your capacity as well.
Excuse me. Okay, I have another question from the audience. Is there a purification step to remove the crystalloids?
It will depend on your, you know, if you have a tag, so if it's an antibody, you can have protein G. I mean, there's a number of ways to separate these, and we don't see the crystalloids every time. So if that does happen, there are ways, you know, ion exchange. If you're not, you know, if you're dealing with something that doesn't have a tag, could easily separate. You know, because you know exactly what the protein is, so you can design your process to remove that, or maybe bind that under certain conditions. So
Okay. And do you have to worry about protein glycosylates? Oh boy, I should have had you read this one. Glycosylation. I think I need to go to your University of Georgia.
Can you say that one more time?
Yes.
I'm not seeing that question for on my.
I'll repeat it, Jen.
Okay.
Do you have to worry about protein O glycosylation?
Mainly we see the N glycosylation. I've not seen the O glycosylation with Pichia.
Okay. So we have another question. What is the makeup homogenizer used for generating 2000 bar for Pichia cell lysis? I'd like to know what you use. Just one comment I'll make on that. It is a, you know, it's not impossible, but it's not a straightforward technology to scale. And I think some of the homogenizer technologies are easier to scale than others. But I'll turn it over to you, Dr. Blum.
Yeah, I mean, the good thing about homogenizers is that they're used to make milk, so they do have a lot of bigger systems. Now in our lab, we do a smaller scale homogenization. So we have it's called the Panda 2000, and it's from GEA. And so that's our 2000 bar system. We also have a constant systems homogenizer. So both of those are going to get up to 2000 bar. And they both have a flow rate of about 9 liters per hour. So you can lyse, typically if we have say 10 liters of material in just a few hours, you can do 3 passes and get, you know, a fair amount of lyse cells.
So we have another question. The maximum concentration of methanol can Pichia bear? And my interpretation of this question would be what concentration of methanol do you start to see significant reduction in productivity or growth rate?
That's a good question. I'm not sure about that. I mean, we typically keep this, fairly low because we have sensors to keep the methanol concentration low. I would say we wouldn't go over maybe a couple of percent of the total volume in methanol, and the cells will use it up fairly rapidly. So we're really feeding on demand. So I think for that, you don't want to get too high, but just a couple of percent would be okay. And definitely use the sensors to maintain your methanol.
Okay, in your opinion, which chaperone or helper proteins are limited and need overexpression to improve the secretion and yield?
I've not seen Pichia strains like the E. coli. There are chaperone expressing strains, so that is something that I've not seen on the market as far as strains. But you could definitely engineer a Pichia like they engineer the glycoswitch to express a chaperone. So you'd obviously want to pick one that was compatible with that the strain of Pichia you're working with. But in general, I've not seen chaperones employed.
And can we use Pichia for biopolymer production like PHA, PHB?
That is a good question. I know that there are a lot of wild type organisms used for this, so you would definitely need to engineer in the pathway. I think it's possible. I'm not a PHA or PHB expert, but I would, I think a metabolic analysis could be done to look at, improving the Pichia to do that. We actually have researchers at UGA that are experts in strain development, so that would be a really interesting project to work on.
So we have another question. Is the methanol feed rate for production critical, or can it be slowed down to accommodate schedules for convenience? And, comparator might be E. coli, right? Where the feed rate can be very critical in terms of what type of inhibitory products you can get. But I'll do some initial comments on that. So any feed rate can be slowed down. And it's more commonly done to accommodate at larger scales your bioreactor capacity, either for mass transfer of oxygen or heat transfer out of the system. Heat transfer starts to become limiting because your surface area goes down as you scale up. And so that could be a limiting factor. And so you can use feed rate to control those things really quite effectively at larger scales. But I'll turn it over to you for additional comment, Dr. Blum.
Yeah, so thank you. So for us, it's because we have one shift, we would typically change the feed rate to go over a weekend. So it would help us as far as covering the work. But yeah, I mean, definitely you can slow down the feed rate. As a CMO, we're kind of implementing processes that our clients want us to do, but we definitely could consider doing a slower feed rate, and that would, you'd also want to look at temperature and pairing those two together to get an optimal production if you want to stretch out your total run time.
We have another one here. Based on your experience, should we tag or not tag the proteins for purification to get a good production yield? I would say it depends on the application. I know for E. coli, you would never want to try to go through the approval process with a tagged protein, just because of some of the potential deleterious aspects of that and regulatory types of issues. But could you elaborate on tagging or not tagging proteins depending on application and use, Dr. Bloom?
I would agree with not. I mean, if it's going to be a biopharma, then you wouldn't necessarily want to tag that. we've had a lot of success with adding in TEV or other protease cleavage sites to remove the tag. But whether you add the tag or not will depend on how, if other techniques to purify. So we would, if we're not able to tag, then we typically utilize some sort of ion exchange type process. But if your application can tolerate a tag, I would always recommend using the tag. And we have a lot of experience with his tags. And when we remove the tag, we can then repeat the purification, remove the protease, the tag that was cleaved off, and then anything else that bound the first time to the column. So it's really a great way to clean up your protein. So we implement that quite a bit. But it would, as James mentioned, depend on if your regulatory requirements allow or prevent that.
Yeah. For example, if you're doing something for a diagnostic, I think that would be a really good application.
Sure.
For the technology, for example. It looks like we've run out of questions. I'd just like to give one last call for any more questions. But we really appreciate everyone's attendance. And again, we're here to help. If you have any questions or if there's any way that either organization can help you after the call, we'd be happy to hear from you. Any more questions from the audience? Jen, I'll turn it over to you for concluding comments.
Yeah, excuse me, absolutely. Just wanted to thank the attendees and all the questions that you posed. As James said, please let us know if you have any more questions that come up or if there's anything that we can do to help you. I do want to just extend our thank you to Dr. Blum for joining us on this webinar today. It's been very insightful. We hope that you all found it instructional as well. We will send out a link to the recording within the next few days, and it will also be available up on the Windshire website at windshire.com. If you can join us for our next webinar, which is going to be coming up on September 8th at 11 o'clock Eastern Standard Time, we are going to be talking about risk and how to assess and manage it across your organization. But again, thank you all for attending. This does conclude our webinar for today.